All right. So thanks for coming to our July webinar for the Cancer Genomics Cloud. We have a great speaker today. Caitlin Guccione is a graduate of the University of Rhode Island with a bachelor's degree in applied math and computer science and a master's in applied math, but is now pursuing a PhD in bioinformatics and system biology at UC San Diego, co-advised by Dr. Rob Knight and Dr. Kit Curtius. And her dissertation focuses on exploring the mathematical modeling of the cancer microbiome, something I think we don't think about a lot in cancer research. So with that, Ms. Guccione, take it away.
Awesome. Well, thank you so much for the introduction. And I will start by sharing my screen. Okay, so just confirming, can everyone see that all right? Perfect. Okay, so I will go ahead and get started. So yeah, my name is Caitlin. I'm a rising fourth year in the bioinformatics program. And today I'm going to be talking about enhancing microbial insights in cancerous tissue, unveiling the hidden microbiome within an advanced host depletion pipeline. So let's get started. So I want to start off really kind of high level about what is the microbiome, because a lot of people in cancer don't think about the microbiome, and it's kind of a new area. So basically, starting off on the left here, we just have microorganisms. So these are just any tiny organisms living in all kinds of environment. And these usually are made up of bacteria, fungi, viruses. And the microbiome itself is a community of these microorganisms and their role within a specific environment, considering environmental conditions and interactions with each other. And so what most people think of when they think of the microbiome is the human gut microbiome. So basically, we all know or have seen ads at least for the role of the gut microbiome in nutrition and health. So a healthy microbiome often is linked with like probiotics or fiber intake. This causes lower inflammation and a lot of other great things going on there. And a diseased microbiome, this is often linked with excessive sugar, saturated fats, or protein consumption. And this can be linked to a higher risk of inflammation and a variety of other different diseases that have a negative impact on your gut. And what is a little bit lesser known is that the human gut microbiome doesn't just affect the human gut, but can affect a huge range of other things. So starting off, the human gut microbiome has a link to a ton of things that have to do with the brain, things such as appetite disorders, Alzheimer's disease, anxiety, depression, stress, and even addiction. And this is all being newly studied, and this is more of a new area of research, but it is an important area of research to understand how your gut affects your brain. Not only does it affect your brain, but it also has links to cardiovascular disease, liver disease, obesity, diabetes, and a range of other things such as like allergies and inflammation. So what's lesser known as well is that not only is there a human gut microbiome, there's also a human oral microbiome. And there's actually different human microbiomes all over the body. So for example, the human oral microbiome has slightly less microbes than the gut, but it still has a good mix of viruses, bacteria, fungi. And there is a healthy microbiome that is in the oral, that should be in your mouth, as well as the option to have an unbalanced microbiome, which can lead to another entire range of diseases there. And so not only do these take place in your mouth and your gut, as I mentioned before, but they're all over your body. So you have a microbiome in your esophagus, on your skin, a vaginal microbiome, and all throughout the body. And there's actually more bacteria that reside in the human body than actual human cells. And the ratio of microbes to human cells is 1.3 to 1. So by not looking at all these microbes, we're actually ignoring a large part of the cells in our body here. And there is between 10 to 100 trillion symbiotic bacteria in the human body that make over 10,000 different species. So the human microbiome is very extensive, as we can see, and it doesn't just focus on our lovely gut. So you might be wondering why I'm talking about this and the link between cancer and the human microbiome. And on the left here, I have the 2011 release of the cancer hallmarks. So I'm sure most of you know what these are, but these are just basically different parts of, these are all linked to cancer, and they either have to do with the development of cancer or the prognosis of cancer or how cancer may get worse or better. And what's really interesting is that the human microbiome is actually linked to 15% of cancer cases worldwide. And these are usually due to either H. pylori, which is a microbe that causes stomach cancer, or to HPV, which causes a wide range of cancers. But the question is, first of all, are there even more cancers that are caused by microbes? And secondly, are microbes having some further interactions that are not necessarily causing cancer, but having perhaps like an interaction between it? And so this is the question that my colleague Greg Poor asked in his Nature paper that was released in 2020, is are there microbes in the Cancer Genome Atlas, or TCGA? And for those of you that don't know, TCGA is just an extremely large cohort with tons of different cancer types available in it. And so basically what he looked at is, can we pull out some microbes from this data, and what do we find if we do? So at the bottom here is all the disease types that are in TCGA, and these red dots here are the total sequencing reads classified as microbial. So as you can see, it's not a lot that are classified as microbial, but it looks like there's around 2 or 3% that are classified as microbial, depending on the disease that you're looking at. And further down here, he asked the question of, out of these reads that are classified as microbial, how many can we actually define as knowing the species? And we can see that it is less than 1% for some, but this is still an extraordinary number of reads. 1% seems like a low number, but there's so many reads in all this data that there's still a ton to look through. And so what he found is that there's differences in microbes of tumor tissue across different cancer types. So looking here, this is the heat map of classifier performance comparing one cancer type versus all others. And the takeaway from this is that each cancer type does have a unique microbiome. So basically what this data is telling us is, if we look at these ROC and PRs, that the higher they are, the larger that area under the curve is, and the more confident we are that we're able to identify, just looking simply at the microbes in the tumor tissue, whether we have breast cancer or something else, or lung cancer or something else. And he took this a step further, and he also looked at comparing cancer versus normal tissue samples. And we lose some of these here because if we didn't have enough samples, we just went ahead and dropped those. So that's why you see some of these NAs. But you can see that this still worked very well in the cases that we did have available. And then finally, he looked between stage 1 and stage 4 cancer types. And again, we lose some of these here, and this didn't work quite as well as the other methods, but what's interesting is in some cases, it still does appear to work with some certainty. So obviously this is an incredible breakthrough to see that this is working, but once we have a tumor biopsy, it's obviously not super helpful to know what stage it's at or what cancer type it is, because that's pretty clearly been established by the surgeon, or now we have the physical tissue. So that's not really like a groundbreaking result. So what Greg wanted to do next, and is kind of like the final loop of this paper, is look at this in blood. So what he did is he took the blood samples for all these patients and he extracted the microbes out of their blood. And he asked the same question as he did originally, is can we identify one cancer type versus all other based upon the microbes that we see in the blood? And he found that this also worked extremely well, especially in particular cancer cases. So the conclusion he drew is that we can identify cancer type in patients using microbes found in the blood. So this was an incredible study done, and obviously there's still a ton of work that needs to be done to validate this, to do further up studies. But the whole point was that using the Cancer Genome Atlas, which is this really large cohort, we were able to at least get an idea of what's going on here and kind of give further justification for why cancer microbiome is really important and should be further studied. And because of this paper, along with a variety of other papers, and the new released Cancer Hallmarks in 2022, microbes have been added as a potential key factor of something that either progresses cancer or has to do with cancer reduction. Shortly following that paper, Greg released one more paper with collaboration with some people in the Weizmann Institute, looking at not only bacteria, which is what his first paper focused on, but also fungi. And he actually found that both in the blood and in the primary tumors, that adding fungi and bacteria together and looking at those reads gave an even larger or an even more confident prediction model of what cancer type we were looking at. So that was a further validation that the fungi are helpful, but we should also be adding them into this mix. So Greg isn't the only one that's working on cancer microbiome. This graphic here, which is now actually a little bit outdated in 2021, shows a variety of different cancer microbiome studies done across the body. So each of these dots just represents a study, and each of the colors represents the type of study that we're looking at. So the blue one is diagnosis, we have purple is mechanism, orange is prognosis, and red is treatment. And what I find really interesting about this is we do have a ton in colorectal cancer, but we also have a wide range of other ones that we can see here. So we have some that are going on in the brain, we have a ton in the lungs, some in the breast. And so there's a ton of people that are looking at all these different cancer types and how the microbiome is actually associated with them. And I know from being in the field that a lot of people's biggest concern is contamination or mislabeling some of these microbes. And so what the Wiseman did that's highlighted in this study is they actually went through and they looked at different cancer types under a microscope with immunofluorescent for bacterial 16S rRNA. And so what you can see here is everything that's kind of lit up is a bacteria that they've highlighted. So not only are we confident in this, we can physically see that in ovarian cancer, there are microbes actively in this tissue. In melanoma and in lung cancer, we are actually seeing microbes within the tissue tumor and having some sort of role either in progression or at least in the environment of where they're sitting. So to kind of wrap up this summary section, I wanted to just briefly talk about the future directions of cancer microbiome. So Greg and I wrote this review paper that kind of talks about opportunities to improve clinical care within the cancer microbiome. And I wanted to just highlight a couple of them here. So at the top, we have survival prognostics based on gut and intertumoral microbial diversity. So there have been some studies that have shown what your gut microbiome looks like can determine how you react to different immunotherapies or different therapies when you have cancer. And by just changing people's gut microbiome using diet, we can actually improve the outcomes of how they react to the treatment. This is a very easy way to kind of improve people's chances of responding to immunotherapy or other treatment methods, which is awesome. Here to the left, we have cell-free microbial DNA liquid biopsies. And this is kind of the key finding of Greg's original paper of that. Can we determine that a patient has cancer or what type of cancer they have looking simply at their blood? And this is actually being spun off into a company called Micronoma that's being run by Greg Poor and Rob Knight. And what they're looking at here is, can we use this as a diagnostic technique? And they're actually starting with lung cancer because lung cancer can be extremely hard to diagnose. And often, I'm not a surgeon, but I can already tell that if you just look at a lung, we wouldn't want to go in and dissect something from someone's lung if we aren't 100% sure it's cancerous. So this is a potential method to confirm that we actually see cancer and it's not just a different type of tumor that's not cancerous. At the bottom here, we have how drugs and microbes can interact. So on the left here, we talk about how when drugs are given, sometimes microbes can kind of interfere with those drugs. So in some chemotherapies, microbes may find them, for example, delicious and eat all of the chemotherapy, not allowing any of it to get to the actual tumor and help the patient. On the right here, we have the fact that microbes themselves could be used as drugs. And this is an entirely new field of study that I'm not an expert in, but that is extremely interesting and could be potentially very useful. And then finally, on the right here, we have how this can be used for pre-cancer and cancer diagnostics, understanding how the microbiome changes and perhaps being able to change the microbiome to stop people from progressing. All right. So now that we understand a little bit about cancer microbiome and why it's so important, I'm going to talk a little bit about a pipeline that we use on how we actually extract microbial reads from whole genome sequencing samples of tumor tissues specifically. So when we're starting off here, we often start off with human tissue samples and then they go through whole genome sequencing. So we're in a little bit of a unique situation, as in cancer microbiome. Often we have these incredible studies, such as the ones on the cloud, that are just very well done. They have a ton of different data. They have awesome metadata, but they were collected with the thought of human genomics analysis. And so because of that, they did not account for the microbes in those studies, and they weren't focused on that when they were taking the samples. And so what we have to do is we want to take the data that we have access to and computationally remove the human reads so that we can figure out what microbes are actually in there. And so the way that we do this is we use computational removal of human reads. So we start off with our human reference and all of our experimental reads. And then what we do from there is we basically map everything that we can to the human reference, just as most genomic researchers would do. And then we just keep the leftovers. So anything that doesn't map, we're like, this could be microbial. Let's look into that further. And then we go through a further pipeline that asks the question, we have a microbial reference. There's a variety of different ones that we use. And we basically, each of these dots is a different microbe, and they each have a unique genome. And so we take our remaining reads, hopefully there's more than 2, and we map these against the genomes and try and figure out which microbes we have. And from that, we get a taxonomy table, which is basically our sample. So sample 1, sample 2, and each of these things here to the left is a unique microbial species with just a count of, okay, we found 2 H. pylori in sample 1. We found 4 H. pylori in sample 2. And we'll zoom into this a little bit later. I know it's a little bit hard to see here. So you may be asking the question, why are you doing it this way? There has to be a better way to get these microbes out. And the answer is there totally is a better way to get these microbes out. But we just don't have access to this data right now, which is why we're working with the data that we have. But I wanted to just quickly talk about like future studies that could be done on how to better be confident about the microbes that we're getting out. So we don't necessarily have to use these human host depletion or these computational host depletion methods. So in an ideal case, we'd have longitudinal cancer cohorts, and we wouldn't just have tissue and blood samples, but we would also have stool samples. Since stool samples have a lot more microbes within them, it would make our analysis a lot easier. From there, there's a variety of different analyses that could be performed. And not only would we get microbes and cancer cells out, but we'd also be able to get potentially immune cells or metabolites out. And there's a wide range of different studies that can be done from here on, figuring out exactly what might be going on and further experimental validation that would hopefully then lead to causal versus, impact of microbial mechanisms in cancer. So this is kind of like our dream study, but we are just at least thrilled to at least have these 2 pieces to kind of get an idea of what might be going on. So now we've gotten our tissue, someone else has sequenced it for us. We've taken it and run this computational removal of human reads. The next step is we need to check for contamination because we didn't run this data, so we need to make sure that nothing slipped through. So if we were running this data ourselves, this is a few things that we would do to check for contamination. So the first thing would be when the tissue was actually sampled, we would take some environmental swabs. So the air, the floor, the scalpels, the patient's skin. We would actually take those samples then because there may be some microbes there that are on the skin, but that get onto the tumor sample that aren't actually in the tumor in the body. This is also true even when you're taking whole blood collection. So taking a skin swab of the control when we're doing things like that. In the wet lab itself, there's some protocols that we can use to check for contamination. So although we try and be very clean in the wet lab, microbes are literally everywhere. So that means that in some cases, there are microbes in our reagents or in our blanks. And so what we can do is create a kind of row where we have all of these reagents, just the reagents in there. And if we're seeing a ton of microbes in here, then we can kind of subtract the microbes we see in here from those in our tissue or in our samples and say that, these were things that were in our lab that came up as contamination. Sometimes we even find microbes within sequencing machines. So there's just a lot of different ways for contamination. And often when you're doing human genomics research, you're not going to just get a lot of contamination from other people because you have so much human DNA that it's pretty clear what's actually there. We just don't always have the luxury of having that with microbiome data. And then finally, there's different sequencing methods that we can use to confirm that we're finding the correct microbes. So shotgun sequencing is the same as whole genome sequencing, which is what we have access to. We can also do something called 16S rRNA sequencing, which basically only only detects microbial reads. So that way we don't even have to worry about host contamination because we're only picking up microbes in this case. Okay, so moving back to our pipeline, sorry for continuing to go back and forth, but so we don't have the luxury of having those. So what we're going to do is we're going to check for contamination computationally. And there are a lot of different ways that we can do this, but one of the best ways we can do this is determining relationships between samples using something called RPCA. So as we started off here, this is our taxonomy table we were looking at before with our bacteria here and our samples here. We can see from a brief look that there's just a ton of zeros going on here, which makes sense because a lot of our samples don't have a lot of microbes, and a lot of microbes are really rare and are only in a couple samples. And then there's some microbes, like as you can see down here, that are a lot in a variety of our samples. So the microbiome field has come up with ways to account for this problem. So in this case, they use matrix completion, so zero values do not influence the actual ordination. This paper does an excellent job of explaining that, and I won't go into all this today, but basically, this is an entire field of trying to figure out this problem of how do we accurately determine what's actually across these samples when we have so many zeros and so much inconsistency going on. So all that we need to worry about today is we have our samples and we put them into this plot. And what this plot looks at here is it's a three-dimensional plot, and each dot represents a sample. So in this case, I think we have around 47 samples here. And how close the dots are in this 3D space represents how similar they are microbially. So for example, these two dots are two samples that are very similar, but this dot and this dot are two samples that are a lot different microbially. So we can form different relationships from looking at this and being like, okay, you know, these are all very similar, these are a little bit different. And this is just one way to visualize that. So what we do is use that to check for contamination computationally. So now I want to actually show you guys an actual example of how this is used in the real world. And we're going to use the Hartwig Medical Foundation dataset to do this. So this contains over 5,000 metastatic human tissue samples that have gone through whole genome sequencing. And this results in low depth microbial reads. And what I mean by that is, first of all, there's not a lot of microbes that we've extracted from these. And secondly, tumors themselves do not have a lot of microbes in them. So if you're looking at the gut microbiome and stool samples, there's a ton of microbes there. Tumor tissue has a lot less. So because of that, we're just working with a lot less data overall, but we're still going to try and see what we can find. So what we did with this data here is we ran it through a host depletion pipeline that we've already talked about. So basically, we started with our tissue samples, someone else sequenced it. We mapped it against HD38 to host deplete, and we put everything into this lovely RPCA plot. So as a reminder, we have, I think, around 5,000 samples here. Each dot is a sample. And the first thing that I just checked in this plot is I was like flipping through and looking, all right, let's look at the sex differences, right? Females versus males. And as you can see here, there is a very clear split between females and males, right? Almost all the males are over here and almost all the females are over here. And we can confirm this with the corresponding p-value of being, there's very clear differences between these groups. So this was really interesting because microbiome does have some differences between sex, but they're not usually this clear, especially when we're looking at tumor tissue. So we either found an incredible new discovery or there's something wrong with our data. So we decided to, check on everything to make sure that what we saw was actually correct. And I, like I said, kind of work at the intersection between cancer and microbiome. So I get to go to seminars that are more cancer related or more human genomics related. So I was at a seminar that talked about this awesome new mapping that is the human reference T2T CHM13 that came out in 2022. And the presenter is flipping through the slides and showing me the Y chromosome and talking about how they have this improved Y chromosome. And I was like, oh my goodness, what does the old one look like? And if you look at HD38, you can see that in the Y chromosome, you have like almost half of it missing. And the reason for this is there's a ton of repeated reads there. It's extremely hard to sequence. So I totally understand why it's not there. But if we're trying to host deplete and we're assuming we're going to take all the human reads by mapping them against this reference, well, all of these human reads we're just shoving into our microbial pipeline. which is problematic because if you have too many reads that are non-microbial into this microbial pipeline, it's going to eventually map some of those as being microbial. So I was like, well, this is interesting. This is much fixed here. So what we decided to do is improve the pipeline and filter against HD38 and then against CHM13 just to make sure we get everything out. So we did this and we remade the plot, and we have a very different story going on here. So we have males and females, the same color contrast, but we can see now we have a very mixed bag between males and females, and our corresponding p-value is very different. So this is just an example of how delicate this host depletion pipeline is and how important it is to check things like this and look through and make sure that the contamination is not leaking through. I actually wanted to take this one step further and confirm that I was actually just extracting male reads. So what I did is I looked at male samples and female samples, and I looked at the number of reads removed with the addition of the CHM13. And you can see that this release not only improved the Y chromosome, it did a couple other things as well, right? They were able to extend to other regions as well. So you can see that in most cases, the females, they lost a couple more reads. One person, I guess, lost a lot more, but for the most part, pretty simple. But these males just lost an extraordinary number of reads. And this is all basically things that should have been mapped to the Y chromosome that were not before. So now we have this improved host depletion pipeline that we're more confident in. And so we can now with confidence go through this pipeline a little bit better. So we have, just as a recap, our human tissue samples, our whole genome sequencing, we have the computational removal, checking for contamination, and then finally we can go back and figure out what we actually have. So I wanted to use fecal microbiome studies to kind of check our host depletion pipeline again, just as another confirmation. Are we getting everything out that we really want to? And so the way that a typical fecal microbiome study workflow works is we have a patient that we often have a fecal sample from. And fecal samples are often publicly accessible because they are thought to contain either very small amount of human reads, or in some cases, they'll do a host depletion here to eliminate some of the human reads. So this is a little bit of a concerning thing for the field of fecal microbiome studies, because we assumed that there are not enough human reads here to do much with. So normally we have these DNA fragments, we take the microbes, and then we go on and do all of our analysis. But this paper came out in May of 2023 that basically found that even with some of these host depletion methods, we can actually figure out who these people are. So we were able to write the news and reviews article associated with it that found, okay, we have these stool samples, we have these DNA fragments. Most of it is microbial in the fecal case, but there's some human stuff, and the human stuff that slips through is able to identify the sex of the patient, the location that the patient is likely from. And in some cases, when we had a match set of, these 300 patients are in the study, we were able to figure out who those people were. And this is all their work, by the way. It was incredible work done. And so what we were really interested in is what happens, like we have this really cool new pipeline, like does our pipeline get rid of this and not allow people to figure out what sex is in these samples? And so we're currently working with them to see if our pipeline removes these. And that would give us further confidence that we are removing a lot of things, and hopefully there's not a lot of human DNA that's slipping through. Finally, another new development, because science always got to keep going, is in May as well, there was a pan-genome released. So now the T2T-CHM pipeline is great, but it's not the current, it's no longer the most up-to-date human reference that we have. And for those of you that don't know, the human pan-genome reference is in this paper, and they basically found 47 pan-genomes, which is across a wide range of different people to try and account for diversity, since different people obviously have different genomes. And this is kind of the year one of the project, but they're going to do further things later on to eventually have over 350 people in this study to try and create the most cohesive pan-genome that's possible. And so currently we've adapted our pipeline a little bit to basically extract in our host depletion, take things from human reference 1 and host deplete from there, and then run it through and host deplete on human reference 2, all the way down to 47. This is effective, but it's obviously very computationally expensive. So what we're trying to work on right now is developing a method that's using these awesome tree-like structures that are also available with this pangenome that basically keep all the stored information together. So, we have this like read that's all the same. There's no need to host deplete by this chunk 47 times, but this chunk, changes. So we might want to host deplete by that depending on the variation. So we're just currently working on that, but the pangenome is a really great discovery that may also improve our pipeline. So finally, the last thing I want to talk about today is the target data set. So this kind of like brings the whole talk together on the Cancer Genomics Cloud and why we're talking about this type of data today. So the target data set characterizes several different childhood cancers. So those of you that don't know, Target contains around 14,000 samples that can be translated into low-depth microbial reads, from what I could tell from what's available online. And there's a ton of various cancer types that are included within it, with the goal to get a better understanding of childhood cancer. And so we have some big picture childhood cancer microbiome questions that we're hoping to answer using the target data set from the Cancer Genomics Cloud. So the first one is kind of the same question that Greg asked using TCGA is, can we see differences comparing one cancer type versus all others using tumor tissue? And then the second follow-up question is, can we see differences comparing one cancer type versus all other using these normal blood samples, as Greg had found before? And then finally, are the microbes represented in childhood cancer similar to those in the corresponding adult cancer? So are we seeing microbial differences between these two cancer types? And then using some additional data resources, we hope to validate these findings across a larger subset of childhood cancers, and then also validating these findings across another data set with similar cancer types to make sure that this isn't just unique to whatever data is available through Target. So as a breakdown, we start off with our target samples, and a large chunk of this is RNA sequencing. And RNA sequencing is great, but it often has a lot less microbial DNA with it. So we decided to start with the whole genome sequencing first. So the whole genome sequencing has 576 BAM files across 6 childhood cancer types. The reason that we lose some numbers is some of them become so low that we can't look at that subset. So that's why our numbers kind of drop as we go through this flow chart. And basically, these whole genome sequencing samples can be split into 3 types. That is blood-derived normal, primary blood-derived cancer bone marrow, and primary tumor. And so, as we always need to do, we need to check for contamination first. So what I did is I took all these whole genome sequencing reads, I host depleted them using CHM13 and HG38. And the first thing I checked is that the sex looks okay. And we can see that there's a pretty good intermixing between males and females here. So that's great. The next thing I checked is, as I said, flipping through a lot of these different categories is helpful to make sure we don't see splitting by something else. And what I noticed here is that we see different clusterings between the blood normals, the bone marrow, and the primary tumor. And that makes a lot of sense, right? These are very different environments. So comparing these bone marrow samples to these primary tumor samples is probably not a great idea. So what we decided to do, and what Greg had done in the past as well, is kind of separate these out by their different tissue types. So we decided to start by looking at the blood-derived normal because we had the largest number of BAM files there. And so that's where we started. And this is all very preliminary data, so there's nothing groundbreaking here to start, but it is interesting just to see it at the very beginning. So we have 3 different cancer types here, and we can see that we are seeing a little bit of clustering happening. So it seems like the green dots are all right here. There's a good mix of red here. They're a little bit up here as well, and the blue kidney samples seem to be up here. And we do see a corresponding p-value separating these two. So this is by no means like a groundbreaking result, but it was promising to see at the beginning that we are seeing separation. And so we have a ton of more research to do on this. So basically, our next steps include finishing the analysis of our RNA sequencing data, which will give us a lot more data to work with and a lot more dots in our plot. And then combining the analysis of the whole genome sequencing and RNA-seq data as was done in Greg's paper. And one thing that Greg's paper did that I didn't highlight at the beginning is basically there's ways to account for differences you see across different sequencing technologies or across different samples like we do here, where we saw clustering basically between, each of these subcategories here. And so the way that you can deal with this is by using a bunch of different methods to kind of remove data that is causing data to split between whole genome sequencing and RNA-seq, and Greg's paper does an awesome job of outlining that. So we will also be doing that as well, but it's good to look at them individually to start. And then from there, we hope to run random forest machine learning models to answer our original questions that we had talked about at the beginning. So can we see differences comparing one cancer type versus all others using tumor tissue? And can we see differences comparing one cancer type versus all others using normal blood? And then finally, as I said before, we hope that we can identify if these microbes are represented in childhood cancers are similar to those in corresponding adult cancers. So we can then match a lot of these cancers up with things that were found in TCGA and see, what microbes are at the top of this data set and what microbes are at the top of that data set, and determining if there's similarities or differences between cancer types there. So I wanted to wrap up by talking about kind of next steps for the cancer microbiome. So this is just another graphic in one of Greg's review papers, but I just want to highlight again, like how important the cancer microbiome can be for this field, starting off with diagnosis. So we can use cancer microbiome to diagnose particularly lung cancer, is what they're looking at right now. Not only that, but prognosis. Cancer microbiome may determine how well people react to things. There's an entire field on cancer microbiome and the therapies associated with modifying different things to allow them to be used as drugs. And then finally, with remission and checking patients to see if we have detectable diseases or not. And so I really appreciated coming to this talk in particular, because by just using some of these, getting a better understanding for cancer microbiome, and also when we are publishing future cohorts of large data, just understanding kind of what protocols can be in place to check for contamination and encouraging some of these to be taken in the future would be an extraordinary thing to kind of move this field even further forward. So for example, if we had access to some of these things here, which are just a couple more samples when they're being taken, we could have a lot more confidence in the samples being done in cancer microbiome. And we can have a lot more studies that focus not only on the actual genomics, but kind of pairing those two things together to get a really big full picture of all the data that we have available and everything that could be going on with our patients. And so I finally wanted to acknowledge both of the labs that I'm a part of, the Qualitative Cancer Control Lab with Dr. Kit Curtis, as well as the Night Lab with Dr. Rob Knight, and specifically as well Greg Poor, who started a lot of this work with his Nature paper and just being really interested in understanding why the field of cancer is so interested in looking at all these different aspects, but that the microbiome hadn't been looked at and cancers were thought to have been sterile. So Greg really kind of started this for our lab in particular. And then I'd also like to thank all my data resources and specifically the Cancer Genomics Cloud. So thank you all for your attention, and I'm happy to take any questions that anyone has at this time.